now.
Welcome everyone, and thank you for joining our webinar today. To introduce myself, I am Dr Helen Blackshaw and I will be your host for the next hour, where I will be introducing our two expert speakers and posing your questions to them at the end of the session. To give you some background, I work at ENT Clinical. We are a full-service clinical CRO based in the Netherlands. We were set up with the aim to give dedicated expert support to companies looking to run trials across ENT, hearing and balance. We work closely with CILcare, the world's leading R&D services company specialising in ear disorders. CILcare are based in France, the US and most recently Denmark. They have state-of-the-art laboratories where they run comprehensive R&D programmes. The complementary nature of our two companies provides the opportunity to achieve our similar missions, which is to accelerate the development of novel therapies to treat hearing loss. And together we can achieve this because we cover the preclinical and clinical stages of the translational pathway. So that brings me to the topic in question today. How do you make a success of your clinical development programme in the hearing field? For example, how do you choose your animal model for the preclinical work? Or when do you start thinking about your relevant patient population for your clinical trials? Our two speakers today will cover some of the key points for consideration when developing your novel hearing therapeutic research programme. Before I move on to our first speaker, I would just like to remind you to please post your questions as you think of them into the Q&A box on the webinar screen. I will then be posing these questions to the speakers once both presentations have been given. To introduce our first speaker, I would like to welcome Dr Wahid Awad from CILcare. Wahid is the Chief Business Officer at CILcare, where he brings more than 15 years of international expertise in the life sciences domain. Wahid will open today by talking to you about the design of preclinical studies. Over to you, Wahid.
Thanks a lot, Helen, for the intro. Hello, everyone. Really glad to be part of this first webinar. Hopefully it will be followed by a series of more specific webinars in the future with ENT Clinical. So during the first part of this webinar, I will be speaking about the preclinical aspects of drug discovery for different hearing disorders. So I would like to start by saying that it's quite important to have the clinical indication in mind when you start the preclinical phase of your drug discovery, or before you start them actually. So this should also include thinking about the route of administration that you are considering in the clinic, as well as the endpoints that you are interested in reaching, of course, in a clinic again. So after you are done with all the pre-formulation work, which usually takes some time, especially if you are targeting local administration and you need to perform transtympanic formulation for that. And when you're sure about the bioavailability of your drug in the inner ear through all the PK and bioanalysis studies, then you would need to select the best efficacy model for your indication of interest. So this also includes defining, of course, the drug regimen, the route of administration. So is it going to be systemic or local? And then if you choose to administer locally, is it transtympanically? Is it into the round window niche or via the intracochlear injection? So lots of decisions here to make. And of course, the choice of the most translational readouts. But here I would like also to say not only translational, but also what we can call complementary readouts, because these could really also be very informative readouts. And this is usually followed by, of course, the autotox and general GLP tox studies, which we will not be able to cover today, but this could be also a subject for another future webinar. So, and this is a slide where I will stay a little bit for some time, so be patient with me. So here I just have 6 major indications that you could consider, you could target. So at least these are the indications we have established models for at CILcare, and we have the expertise to work with. And I'd like to start from the left to the right. So the first one from the left here is the age-related hearing loss, or also called presbycusis. And for this specific indication, we can use the SAMP8 mice model. So this is a senescence accelerated mouse strain. It's a mutant, but it's not genetically modified. So It's usually associated with high oxidative status, and it's usually represent a model for accelerated age-dependent disorders, which includes, in this case, hearing loss, in addition to cognitive impairment, where we can actually measure both of these. So usually the specificity of this SAM8 mice strain is that it exhibits a shortened lifespan. So this is really less than 1 year old, so that's about 11 months. And usually the hearing loss is observed at the age of only 5 weeks. And this is why this model is usually widely used in drug development targeting cognitive impairment. So I'd like to go for the second indication here, and it's tinnitus or tinnitus, ringing in the ear, depends how you would like to call it. And for that, we have models in long events rats. And we actually have 2 models for this specific indication. The first one is a salicylate-induced tinnitus or salicylate-induced model. And this is where we use it usually as a POC, a proof of concept, because the high dose of salicylate induces reversible tinnitus, and it can be used as a really quick model to see if you can pick a signal that your compound has some kind of efficacy before you go for the more clinically relevant model. In this case, it's the chronic tinnitus that is induced by noise or noise trauma. The next indication here is noise-induced hearing loss, and this could be either permanent hearing loss or transitory or temporary hearing loss, and this is usually translated into ABR, so either temporary structural shift or permanent structural shift, and this is usually depends on the characteristics of the acoustic trauma. And we can do that. We have established models in rats and guinea pig, and we're also establishing a third model in mice, synaptopathy model. So the next indication is drug-induced hearing loss. Really important, specifically when you talk about cisplatin and antibiotics like aminoglycoside, especially gentamicin. So these are very important models also that we have. And some people actually use the cisplatin-induced hearing loss in Western rats as a POC, as a proof of concept, to mainly assess the efficacy of their compound on hearing loss. And then there is a genetic deafness, and to target that, you would need to use a gene therapy model. And the first thing that you need to do in this case is in a gene therapy program is to choose the virus particles that you would like to use. And so far, the most common one that is being used by most of the leading companies in this field are the adeno-associated viruses. And we can do that either in rodents or also large species like mini swine or sheep. And then last but not least is otitis. So otitis is the most frequent type of ear inflammation, and we only focus here on otitis media, which affects the middle ear. It's really a very common type of ear infection, especially in children. And at CILcare, we can assess that the effects of drug candidates on otitis media in chinchilla. So next slide is here going at very important here to say that it's actually the message of today, which is following the screening and all the POC studies. It is quite important to design an efficacy study that mimics the clinical situation, mimics what you would like to see in the clinic. For example, if in your program you can start with a POC study, so in this case you don't have to have the perfect formulation, and it can be probably simpler in respect to the administration regimen. So that means probably 1 dose, 1 formulation, and for a short period of time. Which also again means it's not exactly reflecting the clinical situation, but also gives you really meaningful data here that you can use later on to fine-tune the efficacy study design, that the main study that you would like to conduct. And again, I would like to give an example here of the noise-induced hearing loss, which is really harder to apply in respect to a protective approach from a clinical standpoint, at least so far and until this moment. But what you can do is that you can administer your drug within a short window after the noise trauma. So maybe you can, by doing that, you can save the hearing function, as you can keep the hearing loss machinery at bay, if I can say that, which again means before the onset of the hearing loss that is usually caused by either hair cell or ribbon synapses damage. But then if when you talk about a curative or regenerative study or approach that maybe is more clinically relevant, again, you probably need to run a longer study so we can actually have the time to see the effect of your drug, and maybe you need to do multiple administrations and try multiple doses to be able to see the effect of your drug candidate. So here for the readouts part, I would like just to say that it's important to have at least two of the major readouts that we recommend, the translational ones, which are the ABR and DPOAE for all different types of models actually. And yeah. So this is Animal Farm, and I would not speak a lot in this slide. I just wanted to say that it's important, it's recommended to do all the preclinical package in the same species. So this is starting from all the way up to from PK studies through the PD efficacy all the way to the tox studies. And we can do that at CILcare. We can do that either in a small species like rodents, and also in big animals like, as you can see in the right side of the screen, the mini swine and also sheep. So it's very important to consider which readouts to include in the efficacy study, and of course the autotox studies. But it has its own regulatory requirements. It's quite simple, and we can also speak about another webinar. But if you can look at the left side of the screen here, you could see all the in-life non-invasive readouts that we use in our preclinical models, and can also be used in humans because, as I said, they are non-invasive readouts. But then again, in addition to these main readouts, it's quite important to include some complementary readouts that you can see in the right side of the screen here, because these measures, these readouts, can actually help you to better understand the effect of few compounds, especially if you're talking about cochleograms. You know, that's a major readout to count the hair cells. Next slide. So in this slide, I just would like to say that it's fundamental to have robust and validated and reproducible models, not only in the case of hearing loss, but that's quite in general in drug discovery. And in our case, it's one of the main conditions to achieve a validated model is to be able to conduct the experiments in a high quality acoustic environment. This helps to reduce the heterogeneity and the variability of the trauma inducers, especially when you talk about noise-induced hearing loss. But also you need to minimize the variations in animal handling, and also you need to assure randomization and to use proper controls, and that's very important part too. And another condition is to have a high throughput measures. So to give you an example here, I would like to speak about the ABR. So at CILcare, what we do actually is that we can measure ABR on 8 animals at once, 8 animals simultaneously. And this is really useful to shorten the whole preclinical phase of your drug discovery. And of course, this translates into shortening or reducing time to market. But I also would like to give you here an example of what we can call a high throughput model, which is the cisplatin-induced hearing loss that we spoke a little bit about before in Western rat, because this is in only 4 days. You can test the efficacy of your drug candidates that could be involved in oxidative stress, in inflammation, or an apoptosis. So again, to resume in the slide, it's very important to have a high throughput and highly reproducible model. And this usually represents an initial successful step towards a therapeutic intervention in hearing disorders. And to achieve that, you probably need a highly expert team in these matters, especially, of course, in otology and maybe neuroscience and so on and so forth. So here is my last slide, and I'm going back to this first graph that I showed in the very beginning. And really the most important message of today, you can call it the take-home message, which is think clinical development before you engage in the preclinical program, because at the end of the efficacy studies and all the autotox, GLP tox studies, then you would need to prepare the clinical protocol, of course, for IND submission. And at this stage, you need both the preclinical and clinical experts to work together. And that's why from this point forward of the webinar, I would like to pass it to Helen, and after that to Professor Anna Schilder to speak about the clinical aspects in drug development for hearing disorders. So thanks a lot for your attention. Please don't hesitate to contact me. You have my email here, and I will be happy to answer any question also during the Q&A. Over to you, Helen.
Thank you, Wahid. Thank you for talking us through the preclinical aspects for consideration. I can see lots of your questions have come in regarding the models just discussed, so I look forward to hearing your thoughts on those later. Now we move on to our next speaker from ENT Clinical. Professor Anna Schilder is an ENT surgeon and a professor at University College London, where she leads the UCL/UCLH Translational Hearing Research Programme. Anna is a hugely experienced clinical trialist and heads up the Scientific Advisory Board at ENT Clinical as our Chief Scientific Advisor. Today, she will give you her insights into considerations for the clinical testing of novel hearing compounds. Over to you, Anna.
Thank you very much, Helen, and thank you, Wahid. Well, this slide shows that we often see the translational pathway as a horizontal process running from discovery to clinical implementation. But as Wahid has just highlighted, the preclinical and clinical components of this pathway are actually really connected throughout. And ideally, this is reflected in your translational strategy. For example, the screening of compounds is very much guided by clinical need and opportunities. It is informed by what we know about these compounds, for example, in other disease areas. And one of those examples is our REGAIN trial, where we tested the local application of a gamma secretase inhibitor in patients with hearing loss. And this particular drug was originally developed for oral use in patients with Alzheimer's disease. When it was discovered that the mode of action also played a role in inner ear hair cell regeneration, the compound was taken back into the lab, where indeed it was found to promote hair cell regeneration and thereby hearing improvement in mice that were deafened by noise. And the company that was set up to take this discovery to the clinic, they approached me very early on in the process as a clinician and a trialist to help them put a team together to develop proof of concept. And in our case, this led to a large EU grant and an international phase 1 and 2 trial. And in this project, all steps towards developing proof of concept were approached as a collaborative of preclinical and clinical experts. When we were about to move from the lab to the clinic, we had intense discussions about the design of the trial, which in fact all started with what is the right time to translate? Do we know enough to move to approvals and clinical testing, or do we need to validate the findings of our mouse models into another animal model? And from this followed discussions on the population of the trial of the population to trial the drug in. Do we start with testing for safety in healthy volunteers, or do we start with patients with hearing loss? And 5 years ago, there were concerns that regulators may not approve testing for safety of IT injection in healthy volunteers, because these injections, these transtympanic injections, are uncomfortable and theoretically may carry a risk for hearing. And at the time, it was felt that patients with hearing loss that could potentially benefit from these injections would be more appropriate candidates. Including patients in your phase 1 trial also has the added benefit of being able to potentially combine elements of phase 1 and 2A trials, and that means that you could pick up an efficacy signal earlier on, which would allow you to move more quickly to the next trial stage. But over the last 5 years, we've learned a lot from recent trials of novel hearing therapeutics, and these trials have shown that intratympanic injections are safe. So what we see is that more and more companies move to healthy volunteers for their phase 1 work. When we then move to testing in patients with hearing loss, we need to make decisions which population to target. And of course, that is primarily driven by preclinical data and by the therapeutic approach. But at this stage, clinical input is really key. The question is, how often do we see a patient with a particular type of hearing loss? Where do we find these patients? As a company, one may want to choose a population with a very narrow set of criteria. For example, Meniere's disease with an X number of attacks over an X number of months, or we may want to include patients with sudden onset hearing loss within a few days of symptom onset. But when we ask clinicians how often they see those patients, they tend to overestimate numbers and underestimate the challenges of recruiting these patients to trials. As a company, one cannot really afford any surprises at the stage of trialing, and therefore you really need clinicians in your wider team that do know about trials and about its complexities. Another important area for a joined up preclinical and clinical approach is how we deliver our compounds safely and effectively. And of course, ideally, we would deliver all hearing drugs by a tablet and assess its bioavailability by a sample of inner ear fluids. But unfortunately, when we target the inner ear, we encounter all sorts of barriers. And this means that we often have to rely on local delivery by an intratympanic injection or even by a surgical approach, and on bioavailability based on PK studies in preclinical models. And Wahid has just discussed the main consideration and the perspective of CILcare on how to approach that stage best. What we need to realize is that preclinical inner ear PK and toxicity is highly specialized work, and it's done in only a few labs around the world. And it's important that these labs collaborate with clinicians for successful translation. For example, when we make decisions on formulation, the vehicle for a new compound, it is important to discuss with clinicians because they have growing experience with intratympanic injections, and they have learned what solutions patients tolerate best and what viscosity works best for them when they give the injections. And it's important that this type of knowledge is taken on board. When we move to the clinical trial protocol, important decisions need to be made about the dose escalation strategy and whether we use a placebo. And again, we're learning rapidly from the ongoing trials the pros and cons of various approaches. And importantly, how do we identify the population that is most suitable to the approach that we are testing? I think as a field, we are well on our way to developing precision hearing medicine, but the big question is, are we ready for precision diagnosis? We have made good progress in site of lesion testing in the lab, but many of these advances have not been translated to clinical settings yet, so they may not be readily available for your trials. We always say that sensorineural hearing loss is not a disease, it's a symptom of a wide range of underlying pathologies. And when embarking on a clinical trial, we need to decide how we're going to narrow this range of pathologies, and of course ideally to one, to increase our chances of success. And these diagnostic challenges, they translate into the outcome measures or the readouts for our trials. what is the best toolkit to use? At the moment, there is no international standard. FDA has recently provided guidance that testing in hearing trials should include pure tone audiometry and some form of speech audiometry. But the question is that enough? And Wahid has just shown the wide range of hearing testing that is done in their preclinical models, which includes OAEs and ABRs. And of course, we need to make decisions whether we include these in our trials as well. From an academic perspective, of course, I feel we do, because we want to gather knowledge. But how relevant is that for a company that primarily wants to move forward, pick up an efficacy signal, and move forward with its program? Another complexity is in our field is the wide variation in protocols and equipment used for hearing testing across sites and across countries. And this is particularly true for speech understanding in quiet and noise, where testing those across languages, across countries, adds another challenge for anyone working or investing in this field. So there's a lot that we need to do as a field, and we definitely need industry standards and white papers. But there is more than that, I think. We have made major progress in our field over the past 5 years. We've learned so much. I strongly feel that we need to make the most of those learnings. And as preclinical and clinical experts, we have a responsibility to share. Of course, we need to respect our NDAs and CDAs between companies and institutions. But I feel that by only sharing what we've learned, we will be able to design and deliver better trials, move our field forward, and make these new hearing treatments available to our patients as soon as possible. And Helen, I think you're going to reflect on this a bit more.
Thank you, Anna. Yes, I agree that sharing experience really is crucial to the success of clinical trials. So the first point I want to expand on today is the role of the clinical development team. As you highlighted at the beginning of your talk, Anna, it really is about combining the right team as early as possible in the process of translation. Coming from a CRO perspective, we often speak to companies who have a lot of in-house experience covering drug development, the therapeutic area, and preclinical scientific expertise. In the early stages, it is about looking for the team members that perhaps are missing. For example, a clinician with expertise both in the otology field and with experience in running hearing trials. Often there is in-house expertise in clinical trial operations, but if that experience comes from another field, it may be ideal to involve a project manager, for example, with experience of conducting hearing trials and working with the relevant investigators, and to include them as early as possible in trial discussions. It is then about taking each of the elements of trial design and protocol development you have discussed so far, Anna, and assessing how to put them into practice. So bringing in that operational expertise and networks of sites that may be able to run the trial in question. With the field evolving so rapidly, it is ideal to find an operational partner who is looking to work together over time across a pipeline. This will enable the specific hearing knowledge and lessons learned from each phase of clinical testing to be brought forward to the next. Through this involvement of the operational team, it enables practical elements to be considered early on, saving the need for redesigning the trial at a later stage. For example, when discussing the patient population, it is good to talk through where a company may want or need the trial to be run, in terms of the funding behind the trial, or the countries of interest to a company, and discuss if those countries will be possible when taking into account the operational aspects, such as where these patients can be found in those countries, the differences in presentation or treatment pathways in the countries of interest, together with the relevant regulatory timelines and associated costs. It becomes a balance of those desires, requirements and practicalities that enables effective country selection for a hearing trial. Feasibility and selection of sites is something that is obviously very specific to the area of hearing loss being investigated, whether that be ototoxicity, sudden onset hearing loss, or hearing disorders with a genetic origin. With clinical trials in many of these areas being so new, selecting the sites that not only have the available patients, but also have the capacity, the confidence and the capability to run that type of trial is hugely important. And as Anna mentioned, with many phase 1 studies being run in healthy volunteers, it is important to think about the practicalities of running trials not in a patient population. For example, thinking about whether specific hearing trial requirements can be found for early phase trials, such as the phase 1 unit needing to be close to an audiology department to be able to measure safety in terms of hearing, and whether such phase 1 units have the ability to conduct overnight stays for first-in-human trials. When running trials in phase 2 and beyond, especially in hearing disorders where patient numbers may be low, you may need sites to be distributed across countries to achieve sample sizes. It is then important to consider if the audiological equipment and protocols used at the different sites are equivalent, and so if the data being produced is going to be comparable. For example, in the case of speech testing, you not only need to select a speech test that is comparable and validated in the languages in question, but also explore the experience of the local staff in conducting those particular tests. In some sites, speech tests can form part of routine care, but may not actually be conducted in-house and could be outsourced. This leads me on to an important part of any trial, the training of the site staff. CROs can work closely to provide relevant training for the type of trial in question and provide that vital two-way link between the sponsor and the site to ensure the knowledge and expertise is of a level to make the trial a success. This requires elements of audiological and data collection training from the CRO, as well as education in communication, to ensure these trials are presented correctly to patients by site staff who are most likely very new to running hearing trials. Overall, this partnership and building of operational and site-level knowledge will pave the way for each subsequent clinical trial and work towards ensuring a successful future for hearing therapy development. So that concludes our 2 talks for today, and hopefully you have gained information relevant to the testing of your novel therapeutic across the translational pathway. I would now like to ask both of our speakers to join me for the interactive Q&A session. If you have not done so already, please type in your questions in the Q&A box on the webinar screen now. So let's kick off with the first question, which is for Wahid. Wahid, are the readouts you are using at CILcare translational? Are they being used in the clinic as well?
Thanks, Helen. That's a good question. It's also a very valid question of today's webinar. And I'd like to start actually by saying that yes, the short answer is yes, but I would like to give as an example of noise-induced hearing loss. So this is one of the very major important indications that we also spoke about today. So usually noise-induced hearing loss can be temporary or permanent and can usually affect one ear or both ears. For example, temporary hearing loss doesn't induce visible damage of the hair cells, but actually causes considerable damage to the ripple synapses, which leads to synaptopathy, what we also call hearing loss. But in comparison, permanent hearing loss causes damage to the hair cells itself. So if you look at what is being done in the clinic, how they diagnose that, it's usually a pure tone audiogram, then there's speech recognition, and of course autoacoustic emissions, and ABR, auditory brainstem response. That is usually also followed by wave 1 analysis to identify any synaptic damage. If you remember what I mentioned today at one of my slides, you could see that you would see at least 3 of these four readouts that we actually do routinely at CILcare. So that's the ABR, the DPOE, and also the wave 1 analysis. So yeah, so that's at least 3 out of 4 of these readouts we do routinely in our animal models.
Thank you, Wahid. Okay, I have another diagnostics question here, and this one's for you, Anna. What is the role of genetic testing as a diagnostic workup in trials?
That, of course, depends on what therapeutic approach you've chosen. I think if you're developing a gene therapy for monogenic hearing loss, it's a given. But I think also in other types of sensorineural hearing loss, where we're not entirely sure what genetic predisposition plays a role, it is more difficult to make decisions whether or not to do genetic testing. I think what we tend to do is we try to exclude a clear genetic cause of hearing loss through our medical history and through our routine clinical testing. And at the moment, I think we don't have enough knowledge yet about the exact role between genotype and phenotype to include meaningful genetic testing in many of our trials. But what I would recommend is potentially to take a sample and store for later analysis when we have more insight into those genotypical, phenotypical correlations.
Thank you, Anna. I completely agree. If we move on now to look more at drug delivery, I have a question for you, Wahid. How can you be sure in your animal models that the drug has reached its target organ in the case of targeting the inner ear?
Yeah, so well, as you all know, I guess that the ear organ is quite complex, and reaching the inner ear is also challenging. And as Anna said during her webinar, that there are few labs in the world that can actually do that, that have the expertise to do that. And in the case of CILcare, we did actually tens of studies doing this, all the PK and inner ear administration. And then what we usually do is we administer the drug, and then this could also be systemic or local. So systemic, that means could be oral, could be IV, IP, subcutaneous, but also the local ones could be so the local is into the middle ear, and this could be transtympanic, intracochlear, intramolar, or into the window niche. And I would like to focus here on the transtympanic delivery, which could be performed by different types of formulations, which could be really hydrogels, so solutions or suspensions. And you would need to have really solid expertise working with all these different types of formulations. So what we usually do after we administer the drug, we sample routinely the blood, the plasma, and of course the perilymph, which is the inner ear liquid that contains the actual hair cells. But also we can sample the whole tympanic bola or CSF or the brain. I mean, not or, but as well as, and we can even sample the specific parts of the brain. Especially this is quite important if you are looking into tinnitus, but not in the case of the inner ear. But also I'd like to mention that it's challenging because the perilymph, which is, as I said, the inner ear liquid, is really small in volume. So it's being done ex vivo, and it's usually a few microliters. So this really depends on the animal species you are working with. So it really requires a very special expertise and extra careful to be able to deal with such a very small volume. And of course, all this work is followed by the bioanalysis work.
Thank you, Wahid. Perhaps, Anna, you can comment here from the clinical trial side. What is possible in terms of PK sampling of the inner ear within clinical trials?
Well, I mean, we see some companies already taking a sort of creative approach. For example, administering a drug in patients that have already been selected for cochlear implantation, or you could even think about stapes surgery, where you deliver the drug briefly before the actual surgery, and during the surgery, when you open the inner ear, you take a fluid sample, and that can give you an estimate of whether the drug can reach the inner ear. I think a lot of people working in this field and having done a lot of preclinical work are actually quite optimistic about any drug that you administer to the middle ear, being able to diffuse one way or another into the inner ear. But we're learning rapidly.
Thank you. So here we have a question about location of performing preclinical animal studies. So Wahid, whereabouts is it in France, the US or Denmark that you perform your preclinical animal work?
So we do actually have 2 labs, one in the US in Lexington, Massachusetts. So where we have a partnership, an alliance with another CRO called CBSET. They are linked to the MIT, and we mainly started to do the GLP autotox studies, but now also we can do all different types of models that we also do in the headquarters in France. And we do this work in France in Montpellier, that's in the south of France. In Denmark, it's still so far, and Paris, it's still more of the business where we do business development. At the moment, there's nothing going on in respect to laboratory work in Denmark. But yeah, we can do all the work in France or the US, and this is really good for us because it can help in reducing the lead time, and then it gives a choice for our sponsors to choose if you would like to do the work in Europe or the US, especially if it's like a US government grant. Maybe they prefer to do the work in the US. There are some specificities also of some of the models, like so far we don't do tinnitus in the US, we only do it in France. And as I said earlier, like the GLP autotox studies, we do it exclusively in the Boston area in Massachusetts. Yeah, but also maybe here I can add something else when it comes to where we do the work. Recently, we have been actually also going to the places, to the facilities of our sponsor to do auditory work when we cannot host these specific animal species in our facilities. So the latest example was dogs. So we have been working with 2 animal health companies. So we have been sending our equipment and our experts to the facilities of our sponsors to do the auditory studies, to do in their facilities. We can also do that actually in a third-party facility, like another CRO that may be able to also host species like, you know, dogs and cats that we cannot host, but we know how to work with. Sorry for the long answer.
Thank you. I noticed you referred to some animal models there, but do you have any other in vitro or ex vivo models that you could mention that could be of interest to people?
Yeah. So this is something I couldn't also mention during the webinar, but it's a quite important question because in addition to the in vivo animal models that I mentioned today, we also have an in vitro optic cell line. So this will be actually, we're working on that, and it should be ready in a matter of few months, maybe early next year. We will have an in vitro assay ready. We also have the zebrafish, of course, for screening, and most importantly, we have the cochlear explants. So this is an ex vivo model too that we established in the US, and it's also ready and going. And so this is also could be very interesting for lots of sponsors that are looking for some kind of work to do before they go in the in vivo animal models. So thanks for this question. It's a very good one.
Great, thank you for the answer, Wahid. I can see we're getting close to time now, so Anna, I think I will put the last question to you. So we've had an interesting one asking about the recent results that have been seen to be appearing in the media. How close do you think we are to taking those treatments into the clinic?
Oh, Helen, if I would know, I think I could put myself in the center of this discussion, but the reality is we don't know exactly yet. But I think what is important is to look back and see what we have achieved in the past 5 years as a field, and that has been phenomenal. The progress we've made in the last 5 years, and based upon that, seeing the results we have achieved so far, I think we can be very optimistic, and I would assume that we have the first treatments in the clinic perhaps in the next 5 years.
Fantastic, and I think that's a great point to finish on, looking to the future. So that concludes our Q&A session for today. Thank you everyone for such a high level of interaction, and thank you to Wahid and Anna for your time to answer these really interesting questions. For the questions we did not have time to address, we will be sending out the relevant answers to everyone who has registered for this webinar. I would also like to point out that the recording of today's session will be posted on both the ENT Clinical and CILcare websites, giving you a chance to review any specific areas of interest from today. So I would like to finish today by informing you about ENT Clinical and CILcare's next joint venture. This will be in February 2021 at the ARO conference. This is a conference that will be held virtually next year, and we will be jointly hosting a research and development workshop to further discuss the topic of translation, together with invited speakers from pharmaceutical companies and biotechs, as well as trialists and clinical investigators, all active in the hearing field. Details of the final date and time will be posted on our websites once the ARO agenda has been released, which hopefully should be later this year. So we look forward to you joining us for that interactive workshop discussion. So thank you once again to our two speakers, Anna and Wahid, for your time to attend, and thank you for asking your questions. I will now close this webinar and wish you an enjoyable rest of your day. Thank you and goodbye.